Okay, so hello, good afternoon. I hope I'm audible and that my screen is visible to everybody. So, um, welcome to the live interaction session for today. So today we'll be discussing the topics for week two. We will start off with a discussion of the assignments for week one. And if anybody has any confusion regarding those, we can look into that. Following that, we will go through some of the topics that were a part of this week, and after that, we can look at some sample problems for week two as well. And simultaneously, we'll also be taking up any questions or doubts that you might have, and we'll try to get back to most of those questions as much as we can today. And if there are any further topics that need to be discussed, we can bring them up in the next week as well. So that is the plan for today. So I will start off with the assignment first. So for the assignment questions of week one, just give me. Yeah. So under week one assignment, the first question had given the similar structure of the central dogma as we have already seen. And there we were asked that the picture below, which depicts the flow of information, which is seen inside cells, that is the central dogma, which pathway depicts the stage dependent on the action of DNA-dependent RNA polymerase activity. So like we had discussed in the last class, if you are talking about if you consider a Xerox machine, which takes one paper as the input and prints out a copy of that paper as an output, then that is a DNA-dependent DNA polymerase. Versus if you think of a scanner, which is taking in the information from a paper and is converting it into something else, which is similar, but is not exactly the same, then that job is equivalent to the job which is carried out by a DNA-dependent RNA polymerase, which is carrying out the process of transcription, where it is reading a DNA template and it is producing a RNA transcript, which corresponds to that very template. So that is being shown here in the process, which has been labeled as 7. So that would be the correct answer in that scenario. Yeah. So that was the correct answer. Then in the next question, we were asked that X-ray crystallographic images of DNA that helped Watson and Crick to build an accurate model of DNA were these images were obtained by. So, like we have extensively discussed, it was there in the lectures as well. That while the initial structure of DNA is credited to Watson and Crick for their landmark discovery of the double helical structure, the images that were analyzed by them were not their own product, but were instead studied or were part of an experimental result, which was performed by Maurice Wilkins and Rosalind Franklin, and that certainly is considered to be one of the biggest contributors or one of the biggest scientific disputes or scientific controversies that have come up in the recent past. So that was the answer here, which is Maurice Wilkins and Rosalind Franklin. Um, Please make sure you do not select the wrong answer in such cases, because if somebody only looks at Rosalind Franklin, you could get option 1 as well. But please make sure you see that distinction. Then in the third question, you were asked that Chargaff's rule for DNA structure states which of the following. So we had that there are twice the number of purines to pyrimidines in DNA. So if we remember Chargaff's rule, it basically talks about how the number of purines and the number of pyrimidines should always be equal. The reason being, which we have also discussed in the last class, the reason being that the hydrogen bonds that we see are responsible for hybridization between two DNA strands is because of the bonds that are formed between A, T, and G as well as C. So these interactions or these hydrogen bonds, by virtue of their chemical structure, can only be formed between a purine and a pyrimidine, and never between purines and just between pyrimidines, because of the nature of their structure, because of the potential hydrogen bond donors and acceptors which are present. Right. So as a result, the number of these two chemical entities is always equal. And the other way to denote it would be in terms of their ratio. So you can never have twice the number of purines to pyrimidines. That is structurally or physiologically, structure of DNA can never have the same. Second option was about how adenine and thymine form double bonds while guanine and cytosine form triple bonds. So while this statement is true, it is not what Sharga's rule is stating. Therefore, this option would also not be correct. Again, another place where you need to make sure that you just don't select the fact which is true, but rather what exactly the rule states. So like we have discussed, the correct option would be option 3, where purine and pyrimidines are always in a 1:1 ratio. Then the next question is that breaking of bonds between which of the following nucleotides requires the highest amount of energy. So we were asked about the highest, okay, so the highest amount of energy would certainly be required between two entities which have the maximum number of interactions, right? So if we look at AT or AU, both of them are capable of forming double bonds with each other, whereas GC form three hydrogen bonds with each other, and therefore they require the maximum amount of energy to break the interactions between them. In question 5, we were asked which of the following statements about DNA replication is true. So first was that Okazaki fragments are found on the leading strand. We know for a fact that this is false because Okazaki fragments are a feature of the lagging strand because of the way in which the direction in which DNA polymerase is capable of moving, which is why it is not able to perform continuous DNA synthesis, and instead it forms little repeats or little not repeats, but short stretches of DNA can only be synthesized at a time. Um, Following that, DNA polymerase replicates DNA from 3' to 5' direction, which is again not true. If this was the case, we would have continuous DNA synthesis on the lagging strand and not vice versa. Then DNA replication is, sorry, something is wrong here. I've selected the wrong option. DNA replication is continuous on both leading and lagging strands. This is also not true because DNA replication is obviously discontinuous on the lagging strand, whereas in the fourth option, DNA helicase is capable of unwinding and separating the two strands of DNA. So that is exactly the job of helicase, where it makes sure that before replication can be initiated and the entire replication machinery is capable of assembling there, helicase is responsible for separating these two strands and keeping them separated throughout the process of replication. So that was question 5. Then the feature of DNA replication that we have discussed extensively is that it is discontinuous and semi-conservative. Please make sure that you understand each of these terms, and if there is any confusion, please interrupt and ask me. I'll try to explain it to the best of my abilities. Then in question 7, we were asked that which enzyme unwinds and separates the two strands of DNA. Again, options here were DNA-dependent DNA polymerase, helicase, primase, and ligase. Please make sure that you know the purpose of each one of them. The first is the enzyme, which is responsible for the synthesis of the new DNA strand. Helicase is obviously the correct option because it is responsible for unwinding and separation. Then comes primase, which is responsible for the initial priming, RNA primer addition, which is required for the replication process. And then we have DNA ligase, which is responsible for ligating the short strands of or short Okazaki fragments that are synthesized. It is responsible for connecting those. Yes, Ajay has a question. Please go ahead.
Hello,
Yeah, go ahead.
madam. Instead of discontinuous, can we use semi-continuous?
Why would you
Because
use
one
that?
stand is continuous and another stand is not continuous. Like we are using semi-conservative.
Mm-hmm.
So instead of
See, these are
discontinuous, I think.
right. So I think the reason why these terminologies, and I am speculating that, the reason these terminologies are used in this way is because when you talk about conservation, it means it is called semi-conservative because one of the parental strands is retained and the other is not, right? Versus if you talk about continuity, the word continuous itself means that it is a yes or no situation. Either it is continuous or it is not. Because one strand is continuous and the other is not, therefore the overall nature is said to be discontinuous. Um, but yes, certainly if semi-continuous could be a terminology, it could be argued upon, but I guess this is the nomenclature that has been incorporated and it is accepted across the world. So probably that is what is continued in the scientific community as well. I hope I kind of addressed your question. But yeah, in question 8, we were asked that if a single molecule of DNA or a double helix, it undergoes 3 rounds of DNA replication, how many molecules of DNA are created which do not have any original strand of DNA? So again, like I think we had discussed a similar question last week. You simply draw a double helix. You just draw out questions like this. They are actually very simple. You have to make sure that you understand that the nature of replication will always remain semi-conservative. So in the first round, you will retain both strands, but in eventual rounds, you will get strands which are completely newly synthesized and no parental strand is retained. And effectively, after 3 rounds of replication, you would have 6 DNA molecules, 12 strands, but 6 individual DNA molecules which do not have any original strand of DNA. So that is the answer given here. Then in question 9, you were asked that a nucleotide is composed of which of the following? I think somebody had asked this also in the last class. So that is a phosphate, the backbone phosphate that we talked about, the nitrogenous base, and a sugar, which in this case, if it is a DNA, would be a deoxyribose sugar. So that was question 9. And in question 10, we were asked that the origin of replication is which of the following? So obviously, this is a very direct question. We don't even need to go through the other options. And the origin of replication, usually denoted as ORI, is the site where DNA replication is initiated or where it begins. So these were the assignment questions, and I think they should be easy to tackle. We will come to the sample problems for week 2 as well. But before that, we'll just try to go through the topics that were part of week 2 so that we can get to it eventually. So majorly the topics for week 2, they were centered around transcription and
Madam,
translation.
one more question.
Yeah,
Please.
please go ahead.
Question number 8, you said 12 strands are formed. I thought 8 strands are formed.
Sorry, let me just check. Where is it?
8, 9. How many strands have original? Question number 7 or 8.
Question number 8. Yeah, how many
Uh,
molecules of DNA are created which do not have any original strands of DNA? Right. So
8
you will
strands
have
are
only
formed.
two
Out
parental
of that,
strands.
-2, 6 will be there. That's how I had written 6, but you just said 12. That's why I got
Wait.
confused.
I might have been wrong. Let's just. So if you draw it out, you have 3 rounds of replication, right? You are starting with 4 strands, of which 2 are parental strands. In the first round of replication, from 2 DNA molecules, you get 4 molecules. In the next round, from the 4 molecules, you are getting 8 DNA molecules. Correct?
Yes, madam. So, madam, simple formula is 2 raised to the power 3. You will get
Mm-hmm.
8. Out of that, original stand will be 2. That is to be removed. 6.
Right. And the
Because in,
remaining that you get
in
are
question
6.
also
Yes.
32. 2 raised to the power 5, it is 32. 32 minus 2, answer was 30. That's how I calculated this.
Okay, we can do something. Wait.
Because 3
We
rounds is
can
okay,
just
but if 5 rounds, 10 rounds are given, that will become difficult thing.
wait. Just give me a minute. So, is the whiteboard visible? I hope it is. So let us say we start with this molecule of DNA. These are the 2 parental strands. So when you have 1 round of replication, you will have 1 of each of these strands and 1 each of the daughter strands, or what has been replicated. Then again, in the next round, each of these will undergo replication. So you will have 2 strands, which are 2 DNA molecules, which are purely made up of the daughter DNA, and you will have these copies also being retained. Sorry, it's not very well drawn, but I guess you get the point. Now, in the next round, after that, what will you get? You will still have two DNA molecules, which are each containing the parental strands. So let me just draw it like this. And with that, you will replicate these. Apart from that, each of these will also be doubled. So you get 1, 2, 3, 4, 5, 6, which are doubling to form 12. I hope I'm not wrong. Just give me a second. Yeah, so 1, 2, 3, 4, 5, 6, and you get 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 11, 12. So how many strands are you getting in the end?
12.
Is it clear? You are getting 6 intact DNA molecules, right? Which have the daughter strands only.
But how in question example they had calculated 30? Means 60 means they had calculated 64 out of that they had done minus. I don't know. There must have been some simple formula. Na?
You can certainly put in a formula. You can start with, say, if you just consider the fact that you start with 2 strands where there are no daughter DNA, one round of replication is giving you 4 molecules. You can use the formula you were using, the 2 raised to the power, but I think for clarity, it is always better to draw it out to avoid any confusion. But yeah, I guess you can just getting a call. Yeah, so you can draw it out. But if you want a formula, I can check and get back with you. But
Madam,
I would
original
always
stand will
suggest.
be in anything. Original stand will be 2 only. Na? How this original stand
Yeah,
will be
yeah, yeah,
2.
yeah. So you always need to subtract 2 from like if you only are starting with one DNA molecule, that is also another thing you need to keep in mind. Here it is specifically mentioned that you're starting with one single molecule of DNA, and
DNA.
therefore you need to get the molecules which do not have any original strands. If your question was how many daughter strands do you get, then your answer would be different. Then you would have 14 daughter strands. But you want intact double helices which do not have parental strands anymore.
Okay, thank you, madam.
Yeah. So I'll stop sharing this. Okay, so yeah, like I was saying, so for this extract. Yeah, so like I was saying, the topics for this week are taking us further along the processes that are part of the central dogma. So we had discussed till replication, which is still here, and further we move on to transcription and translation to finally get the so-called functional molecule of the cell, which is the protein. So how do we get till there? First, we look at the process of transcription, which, as we have discussed, is carried out by the enzyme, which is DNA-dependent RNA polymerase. So if you look here, we have discussed this previously also, that within a cell, that enormous amount of DNA can only be contained within the cell because of the extensive packaging that occurs within. So this packaging is responsible for compacting all of that DNA in the form of chromosomes, which can be separated during each round of cell division among the daughter cells, and it can retain the information. It can carry the information across generations. I think a few people are getting logged out multiple times. Yeah. Screen not visible. Yeah, I guess my screen should be visible now. Uh, yeah, so I was talking about this. So the genes and the DNA that are present within the cell, they are compacted. And I think we had discussed this as well in the last class, that there's what is a gene and what is the difference from chromatid as well. So as you can see that the number of genes in the human chromosome approximately are approximately 20,000 genes, protein coding genes are present. But apart from that, we see that there is a large amount of DNA within the cell which is present, and those are usually considered to be non-coding. And these non-coding genes, they have a different set of functions. They are responsible for regulating the function of the protein coding genes, and they have multiple other controlling functions within the cell. While they do not directly form protein, they control a lot of different downstream functions within the cell. So somebody has a question. Please go ahead.
Excuse me, ma'am. Screen is not visible.
Your voice is very low. I cannot hear you.
Screen is not visible.
It's not visible.
Yeah.
Is nobody able to see? I'll share once
No,
more.
I can see the screen.
I'll share it again. And somebody's video is on. Neela Kantam, somebody. Your video is on. Is it visible now? Please check.
Yes, ma'am.
I hope it is visible. Others, please, other participants, please don't keep your videos on. I think that is the reason there was some problem. Somebody named Repal, your video is on. So please make sure you don't do that. Otherwise, it automatically gets pinned and other people are not able to see. Okay, so coming back here, um, we had discussed the number of, in the lectures, it was discussed that the number of genes in the human chromosome that are present, how many of them are coding and non-coding, what is the approximate number of bases in the human genome, which is near about 3 billion because it is an average number. Then the longest gene among them was dystrophin, which is approximately 2 million 2 million base pairs are present in it. And then we finally come to the process of transcription. So like if we just go over the basic process of it, so what happens in transcription is that you have a stretch of DNA which encodes for a but it has a set of sequences which get encoded into or which get transcribed into an mRNA, which is a messenger RNA. And the reason it is called messenger is because it is carrying the message which is present in the DNA further down in the cell to perform necessary functions. So this mRNA is then it undergoes some processing steps which are known as post-transcriptional processing, which includes splicing. And there's another process as well, which I don't think has been elaborated here, which is polyadenylation. So splicing and polyadenylation, which is the addition of A bases or adenines basically at the end of messenger mRNA is carried out. And then that mRNA undergoes translation, the process by which mRNA is converted into a polypeptide chain, which literally means multiple peptide bonds present. Polypeptide is for multiple peptides, which is just the precursor to a protein. Now, when that polypeptide chain undergoes some necessary post-translational modifications and is properly folded, then it forms a protein, and then that protein is capable of performing all of the different functions that you learn about within the cell. So this is the basic pipeline which is followed inside a cell after DNA has replicated, and the next following processes are taken forward as per the central dogma. So here we are basically seeing the image of a transcription unit, and majorly it has some distinct regions which have also been elaborated in the video. So you see that there is a stretch within the transcription unit, which is where you exactly see the interspersed arrangement of introns and exons. Now, exons, as you know, are the coding sequences. Introns are the interrupting sequences, and these introns are spliced out during the process of transcription, and you ultimately get your mRNA, which is required. Apart from this, upstream of the transcriptional unit is the promoter region, which has some consensus or conserved sequences, which are the Tata box and the CCAAT box. And further upstream, when you see that a stretch of DNA is usually interrupted by a kink like this, it means that these sequences are very, very far from the transcriptional unit. Then these such sequences which are present very far but are capable of controlling or capable of regulating the function of a transcriptional unit which is much, much downstream, those are known as enhancer regions. And as you can understand from the name enhancer as well, they are responsible for or they're usually credited towards increased expression or much higher expression of those genes which might be required as per the conditions within the cell. So enhancers, on the other hand, can be located either upstream, which is 5 prime, and or it can be located downstream, which is much, much down the transcriptional unit, and they are capable of controlling the expression from either of these regions in either orientation as well. So these are some basic terminology which were seen, and within the transcriptional unit, there is also some restriction on what gets translated, but we will get to that eventually. So these are the basic terminologies you should know when you are studying transcription. So then there was a video about it. What we basically need to focus on is that the main enzyme or the main protein that carries out the primary function with respect to the process of transcription is RNA polymerase, which is the shorter name for it. The more elaborate name is that it is a DNA-dependent RNA polymerase. So like we have described, it takes the DNA as the template and it synthesizes an RNA, which is capable of retaining and transmitting or transducing the contained information further downstream. So this is the schematic representation of the structure of RNA polymerase. And here you can also see a slightly more detailed structure. You don't need to go into details about it because processes like these are extremely detailed. If you take any molecular biology book, you'll be able to learn that you will see that there is no end to learning about it. Still, there is extensive research which is going on about the process of transcription and translation till date, because there is yet so much that we do not know. But for now, you can see that it has multiple different units which are differently colored, different protein subunits, and all these together, they come together to perform all of the functions which are credited towards the RNA polymerase. And here you can also see the overall process that takes place. So you have the DNA molecule, which will serve as the template in this case. It is separated in this manner such that you can see that there is some enzyme which is or some protein which is making sure that DNA is getting unwinded at one end and rewinded. It unwinds and rewinds at either end, and it opens up a small bubble, which in this case is the transcription bubble. In case of replication, it was the replication bubble. Here it is transcription bubble. And within that transcription bubble, we can see that RNA is getting synthesized by the protein moiety that is present. So what you see here is that on one end, you can see that NTPs, or in this case, you can be more specific and say that they are ribonucleotide triphosphates or ribonucleotides, which keep on getting added to the 3' end of the RNA. They keep on getting added, and what you see is that the polymerase moves from this direction with respect to the template. It keeps on moving, and as it moves, it keeps on synthesizing RNA such that the first exiting point has the 5' end of the RNA, and new nucleotides keep on getting added to the 3' end. And this is the main protein which is regulating this function. Apart from that, you have a number of other proteins which cooperate and make sure that the process runs smoothly. There is a transcriptional unit assembly which occurs. There is a pre-transcriptional unit where there is a initiation complex which comes in. There are specific proteins which are required just to make sure that after this RNA has been synthesized, it can be released from this RNA DNA hybrid. There are many ways of doing it. There is a row dependent mechanism. Row is another protein. There are row independent mechanisms. So a lot of detail is there. You do not need to go into it, but if you're interested, certainly go into the suggested books. All of them have these processes in detail. But for now, we can just focus on the processes that are taken up by the polymerase. So I think after that, mainly it was taken up. We do not have much here. So maybe we can just focus on this image. Right. So after this process of transcription has occurred, so what initially happens is that the RNA polymerase enzyme, it assembles here from the promoter region. It starts transcription and it will proceed in this direction. It will keep on synthesizing mRNA or it will keep on synthesizing RNA. And after it has processed, what the next modification that needs to be done is that each of these individual introns shown here in the pink color would have to be removed, and what remains behind are the green regions, which are the exons. And these exonic regions, they would be ligated to each other by the spliceosome machinery, and what you will get is a complete set of complete stretch of nucleotides, which would be your mRNA that is further taken down for the process of translation. So let me move on to the next lecture. Similar. Yeah, yes, it has the same thing. So that is the purpose of carrying out the process of splicing. Yeah. Okay. So like we discussed, so the process of transcription will only majorly look at the function of synthesizing an RNA from the template, and then that synthesized mRNA will undergo the necessary modifications, which include splicing, certainly, like we have discussed, and also for mRNA, which is actually an identity for mRNA. Like I also previously mentioned, it involves the process of polyadenylation, which is addition of multiple, a long stretch, approximately, I think up to 100 adenines can also be added for some mRNAs. And then or maybe I'm overestimating, but yeah, a large number of A moieties are added to the end of that mRNA, and that specifically is redirected for the process of translation, which is what we'll come to now. So like you can see here that the enzyme that is required for this process is the RNA-dependent amino acid ligase. So while this is a way of representing the function that is carried out by this molecule, it is basically simply known as the ribosome. The main coordinator of the process of translation is the ribosome. But like you've already probably learned from the lectures, the ribosome is not a single protein or a single molecule. It is an enormous complex. It is among the largest protein complexes that have been structurally characterized till date, and it contains a large number of proteins. And also, interestingly enough, what is the main difference between most other enzymes and a ribosome is that it has RNA. While there are other RNA dependent enzymes also, but one of the first examples where RNA was seen to have catalytic activity, you can call it enzymatic activity, but yes, catalytic activity was in case of ribosome. So ribosome is often also understood as a ribozyme because it is capable of carrying out the catalytic processes that are required during the process of translation. So we'll come to that. So till here, this is the same dogma that is being followed. And we are now looking at this step where after the mRNA has been produced, the next step is to go for translation and produce the polypeptide chain, which will then fold into the protein. So like I mentioned, the ribosome is a ribozyme, and the structure of the ribosome was one of the biggest problems that were tackled by the scientific community for multiple decades, for an extended, very, very, very long time. And these structures that you see here, they were resolved much later. And as you can get an idea, even though it's quite difficult to really conclude from it, you can see that it is a very complicated structure. It has a large number of proteins, but you can see there are some specific regions which are colored differently. You can see that there is a subunit which is, if I zoom in, you'll be able to see that there is a differently colored subunit, the 50S subunit, and a slightly smaller subunit, which is known as the 30S subunit. And apart from that, you'll be able to see three different colors as well. So these are basically different orientations while you're rotating the yeah. When if you consider a 3D structure that is undergoing rotation, you'll be able to see that they look different along different angles. So here you can see these three very distinct sites, and these have been labeled here as well. You can see that there is the A, P, and the E site. Commonly, these are known as the active site, phosphorylation site, and the exit site. While that is not exactly credited to be the full form for these regions, that is how they are normally understood. So this, as you can see, is the structure of a 70S ribosome. And if you're not already familiar with it, that is majorly for the prokaryotic, or rather, if I am being a little more general, mostly the extensively studied prokaryotic ribosome is from E. coli. So they have this 70S ribosome, and its structure was studied in extensive detail, as you can understand from here. So while this is very complicated, its simple representation shows that this is the large and the small subunit which assembled together. You can think of it like a cap, basically a cap which sits on say a person, and these two subunits together will then go along the entire length of the mRNA and will produce the cognate protein, which is decoded from that message encoded in the mRNA. And within this large subunit, these are not very distinctly marked sites, but these are just regions within the ribosome which perform some very specific functions. So you have the A site, the P site, and the E site. So the ribosome will move in, like if I zoom in here, this is the mRNA binding site, and you would have the mRNA along this direction, and the ribosome moves towards the right. If you, with respect to the screen, it moves towards the right, and as it moves, it decodes whatever information is there in the mRNA, and in consequence, from the top, there is another specific site which is known as the peptide exit tunnel, or a peptide exit channel, through which the newly synthesized polypeptide can then exit. So that is the basic idea that happens within a ribosome. Um, so like I just mentioned, so the prokaryotic and eukaryotic ribosomes, both of them have been studied, and now have also, their structures have also been decoded and understood. So here you can see that this is the prokaryotic ribosome. Again, let me zoom in. And it is said to be a 70S ribosome. If you are not familiar with it, S here is it's a unit which is, it's Swedberg unit. It is not directly additive, because if you see that individual subunits are 50S and 30S, they do not add up to be 70. Neither do the eukaryotic ribosomal subunits 60 and 40S add up to be 80. But what essentially it is understood from is that when you subject these cell lysates, or these cell contents, when they are subjected to very high speed centrifugation, extremely high speed centrifugation, otherwise known as ultracentrifugation, then there is a unit of mass which is used to understand how heavy that molecule is, which underwent the process of centrifugation, and that is denoted by this particular unit known as Swedberg, which is denoted as S. So that is how these ribosomal subunits have also been named. So from here you can see that there's a 50S and a 30S subunit, which individually, each of these subunits, they also contain some RNAs, which is what we talked about, that ribosome is also a ribozyme. So individually, these RNAs are capable of catalytic activity. The other eukaryotic ribosome, which is much heavier as well, which you can see by the Dalton weight, which is shown here. It shows that it is the prokaryotic ribosome approximately is around 3 million Daltons, versus the prokaryotic ribosome is around 4 million Daltons. So there's a significant difference between the two, which is why their centrifugation, the Swedberg unit, which is credited to them, was also quite different. And in case of the eukaryotic ribosome, you can see that the RNA molecules are also different. So there's 16S rRNA for the prokaryotic ribosome, while there is 18S rRNA for eukaryotic. And in fact, these ribosomal RNAs have a very, very significant role to play as well. I'll not go into details about it, but if you ever try to understand phylogenetics basically means the study of evolution of organisms. Then very often 16S rRNA sequencing is carried out to understand how organisms have evolved from each other. You can look into these processes. These are very easily accessible. But the reason they are used is because ribosomes are such a integral part of any living being that most of the time, even across millions of years of evolution, you still expect a large part of the ribosomal RNA sequences or rRNA sequences to be conserved. So based on these sequences, you can often find how closely two organisms are related to each other. Like, for example, if you have a cat, a dog, and an elephant, if you sequence the RNA, rRNA sequences from each of them, you would be able to find out how closely they're related to each other. So that's a off-topic discussion, but it is very interesting. And if you want to read up more, you certainly can. Uh, yeah. So now we come back to the structure. So like we said that because this was such an extensive problem, which was taken up by multiple scientists over many generations. Finally, when the structure of the ribosome was solved, these are the three main scientists who were credited with this incredible discovery. And it was a landmark discovery because from there, people have understood so much more, not just about the ribosome, but about solving the structure and how much more can be understood. Therefore, these three scientists and you can certainly see that Dr. Venkataraman Ramakrishnan is also an Indian. While he was not a resident of India, he is of Indian origin, and he is among the very few Nobel laureates that we can credit to the name of our country. And Dr. Ada Yonath and Thomas Stites, they are also credited. They all share the Nobel Prize together, and they received the Nobel Prize in Chemistry in 2009. So this was certainly a landmark discovery. And of course, information like this, not just for this course, but beyond this course also for your learning, for any competitive exams, etc., that you might appear for the future, these details are very important. So try and remember as much of those as you can. And what I was mentioning here is kind of indicative of the phylogenetic relationship that you can see. So if you see here that these are different types of organisms and their ribosomes, they are studied in great detail. And for example, E. coli, Trypanosoma, Saccharomyces, Arabidopsis, and humans, all of them have different ribosomes of different dimensions, having different number of proteins, having different sequence conservation. So all of them can be studied to understand how closely each of these organisms might or might not be related to each other. So that is basically the idea. Now we come to more details about the process of translation. So what you need to understand is that there are currently, or not just currently, but till date, what has been understood is that there are 20 naturally occurring standard amino acids. And you can find the names and the sequences of those in any books that you want. What you need to keep in mind is that each amino acid has different ways of being denoted. There is a triplet, we'll come to the code later, but there is a three-letter code for each of them. So like here, you can see this is serine, proline, histidine, arginine, etc. So their shorter names are denoted by these three-letter short forms, and also each of them can be denoted by a single letter. So the letter code of the amino acid is extremely important because from there only you understand what is the genetic code. So the genetic code was another extremely landmark discovery, which also involved another Indian origin scientist who together they understood that if we consider the fact that there are 20 amino acids and they are being encoded for by four different bases. Again, this was, I think, a question that was brought up by somebody in the last class, that are there any other bases that might be present in any organism. So the answer to that was normally no. And because we are talking about RNA, we only expect there to be A, U, G, and C. Instead of thymine, we expect uridine to be there. So these four bases, they can form 64 different combinations because they are encoded in triplets. And these 64 combinations, while present, would effectively only encode for 20 standard amino acids. So that means there has to be some sort of system that is present within the cell such that more than one codon is effectively encoding for the same amino acid, and this property was understood as degeneracy. And you can see that in these cases, like serine is encoded for by so many different combinations, proline also more than one combination, so you'll find multiple examples of those. And apart from these codons which encode for the amino acids, there are also some other special codons, mainly the stop codons, which are indicated by certain specific sequences, which are not encoding for any amino acid, and instead when the ribosome encounters these sequences, it stops or it terminates the process of translation any further. So that is also required to indicate that yes, we have reached the end of the mRNA and no further translation is required. And apart from that, one very important codon is also methionine. So methionine is also responsible for initiating the process of translation, which is often also in case of prokaryotes, it carries a specific modification, which is known as FMET, which is formyl methionine. It basically means the methionine has a formyl group conjugated to it, and that serves as the initiator codon for the mRNA. So these are the common properties of the genetic code, and some deviations, while they have been found, this is the major conservation, and deviations also are mostly with respect to the mitochondria. So within the mitochondria, we see that there are a few amino acids like asparagine, which have a different set of codons which encode for the same. And also the stop codons for mitochondria have some deviation from this commonly observed genetic code. So these are some of the basic properties of the genetic code that we need to understand. So then comes this, again, whenever there's a discovery, there are some very, very dedicated scientists who put their everything into finding out the discovery. So this was another landmark event in which Nobel Prize was awarded in physiology and medicine in 1968. So from here also you get an idea that how long people have been trying to understand the process of translation. So while the genetic code was understood as early as probably 1965, yes, Nirenberg was credited towards this discovery in 65, and he was a recipient of the Nobel Prize as well. But the next discovery that we just previously discussed, which is the structure of the ribosome, took almost 40-50 years for the discovery to take place. That is in 2009, they received the Nobel Prize for the same. So you never know when your discovery might actually become relevant. So research is ever evolving. Now, like I said, there was another Indian origin scientist, which was Dr. Hargobind Singh Khurana, who also, if you are not familiar, this is another, there is a certain fellowship which is known as the Khurana program, whereby students can apply abroad for internship. So this might be interesting to a few people. You can look it up. So Dr. Hargobind Singh Khurana, Marshall W. Nirenberg, and Robert W. Hawley. These are the three incredible contributors to the understanding of the genetic code and how it actually helps in the process of protein synthesis. So if you look at what they basically did, it was such a simple experiment if you think about it, but it solved everything that they were trying to understand. So they basically just they used E. coli cells, and they made combinations where they chemically synthesized poly A, poly U, poly G, and poly C. They started off with something as simple as that, and always the landmark experiment for the same is using phenylalanine. So when they synthesized a long stretch of polynucleotides which just had U, what happened was that the ribosomes that were present inside these cells, when they were taken out and when these ribosomes were used to translate that poly-U sequence, what they used to see was that there was a stretch of phenylalanine residues. Similarly, they used CCC. They decoded what is CCC, what is GGG, and then they started using combinations. So this was a laborious experiment when they performed all possible combinations to find out what exactly is happening, and ultimately they were able to understand that these are each of the different sequences that are being encoded for by each of the triplet codons. So this was the landmark experiment by Marshall Nirenberg. Following that, Holly and other scientists, they had shown that tRNAs are are another key player that are very, very relevant to the process of translation. And tRNAs, as the name indicates, has T, which is for transfer, and its job is, if I put it very directly, its job is to transfer an amino acid, which corresponds to the sequence that is present within the mRNA. So it is another sort of machine which you can, if we really have to draw an analogy for it, say you think of a QR code. So, and your phone has a scanner. So if your tRNA is the molecule which will be able to see that QR code and will be immediately able to link it to the bank to which that QR code is associated. So that is its job, to read the appropriate code and to bring the corresponding amino acid in response to it. So this was just a vague example. We'll get into details about it. I think somebody has a question. Yeah, Ajay, please go ahead.
Madam, sorry, maybe I may be wrong, but instead of triple, it is just from 64 that site. I think only two base pair are necessary for forming any amino acid. Apart
Why
from
do you think
methionine,
so?
every
Mhm.
there are first two they are common. Third, you can replace anywhere in amino acid.
No, that is not actually the case. The case is that the codon is read in a manner. So um, basically, what happens is, what you need to understand is, within the cell, when a ribosome is carrying out the process of translation, think about it as a any zipper, any chain that you might have, the individual beads or the individual crevices, they think of them as each individual base that is being read by the ribosome. Now, as the ribosome is moving along at a time, it will read three bases, and in response to that, it will bring in an amino acid. So that is the triplet codon. The ribosome never reads two bases, or it never reads four bases. At a time, it will always read three. What you are talking about, the conservation of bases at each of these three positions, that is debatable, because the third base pair usually has a little more freedom, where it allows some amount of degeneracy to be present. It allows a little bit of wobble base pairing, which is why, irrespective of what might be the somebody was getting it. Yeah, so wobble base pairing, which allows some amount of flexibility in what exactly is present in the third base. But the triplet codon was the landmark discovery, which was understood that, say, suppose you have an mRNA which has 25 bases, right? Then the ribosome will be able to read three triplets at a time, and it will synthesize, or it will incorporate for each triplet one amino acid. So it will incorporate 8 amino acids, but the last base is something which it will not be able to read. So it will never insert anything against it. And under those circumstances, the ribosome would be stuck. It will not move any further. So I hope you got my point.
Yes, madam.
Yeah. So, yeah, we were talking about tRNAs. Okay, yeah, it's mentioned here as well, the Khorana program. It is an extremely prestigious program. So if you want, please go ahead and read up on it. And it's a very wonderful opportunity. So coming back to the process of translation that we were talking about. So like I said, tRNAs or transfer RNAs have a large part, or a very large role to play in this context. They basically carry out a two-step process. The first is where tRNA charging is done, and the second is where its incorporation or its role in protein synthesis comes in. So during the process of tRNA charging, which is what you see here, what exactly happens? Charging simply means that Okay, so if you think about it, tRNAs are also RNAs, right? So they are encoded for somewhere within the genome. Now, after they have been recognized somewhere in the genome, the transcription of these regions have been carried out. They will never undergo translation because they are again not protein coding genes. They are RNAs. Now, these tRNAs take up a definite structure, which is also something we'll come to very soon, which is known as the clover leaf structure. The 3D structure is known as the clover leaf structure, where the 4D structure is it's like the letter L. So it takes up this structure, and then this tRNA has to be charged with an amino acid so that it can do its job. So that is basically its responsibility. So this charging step is actually one of the most crucial steps. Why? Within the cell, you will have tRNAs for each individual amino acids. I think you can see it here as well, probably. Multiple different like individual tRNAs are present for each of the amino acids because they are all varying in this region, which is commonly known as the anticodon loop. This anticodon loop is the region which is able to recognize the triplet codon that we were just talking about. And therefore, whatever anticodon loop is present within the tRNA, it is an extremely important step to ensure that only the correct amino acid is used for charging of that particular tRNA and not the wrong one. Therefore, the enzyme which carries out this charging process, which is known as amino acid tRNA synthetase, this is also extremely amino acid specific. This enzyme is actually considered to be the most, or if not one of the most, probably the most efficient or the most accurate enzyme present within the cell. Like your DNA, RNA polymerases, damage repair enzymes, all of these are still capable of making mistakes. But amino acid tRNA synthesis it makes very, very, very rare mistakes because its mistakes are capable of causing very dire mutations, very far-reaching consequences, and therefore it is one of the most highest fidelity enzymes that is known to the living world. Again, this could be a question for competitive exams sometime in the future as well. So read up a little more on it. There are multiple reasons why it has this high fidelity. There are some filters which are in place. There are some mechanisms which check that after an amino acid has been incorporated, it is the correct amino acid and not a wrong one. So if you're interested, please go ahead and read up on it. So after this process of charging has been done, like in this example, here you have AAA in the anticodon loop. And with respect to that, you expect that this tRNA should be able to recognize phenylalanine, and therefore it gets charged with the correct amino acid, which is phenylalanine. And after it encounters the same in the mRNA, this phenylalanine amino acid would be incorporated into the elongating polypeptide chain. That is basically the idea behind it. So we were talking about the structure of tRNA. So this is the three-dimensional structure and this is the four-dimensional structure. Please make sure this is a very, very common question in any competitive exam or even general exams for this course as well. So please make sure that the 3D structure is said to be the clover leaf structure because it looks, if you've ever looked at a clover leaf, you can Google it for your understanding if you need. It has three such distinct domains and a short variable loop. So this is the D loop, the anticodon loop, and the T-Cy loop. And this end is the acceptor stem, which has a 3' tail where charging actually takes place. And its 4D structure, on the other hand, is what makes it look like the letter L. So its tertiary structure is L-shaped, while its secondary structure or 3D structure is clover leaf. So please make sure that you do not make a mistake among these two. So yeah, that is the idea. These are short sequences. They are 70 to 80 nucleotides in length. They have clover-leaf secondary structure and they fold into the L-shaped tertiary structure. And double helical stems are present. You can see here. I think somebody had asked about the sequence variation as well. So again, this is a separate study that needs to be carried out, but basically there should be complementary base pairing so that you can get such hairpin structures or such stem loop structures within the tRNA. And this is the acceptor stem, like I discussed, and the amino acid is attached to the ester linkage of the 3' OH group or the 2' OH group of the adenine residues. And here you can see a lot of different modifications can be seen, like psi is present, you get inosine, you get pseudouridine, and you get many different types of variations within the tRNA, but they are not usually that common, and it is restricted to majorly the tRNA under such circumstances. So here you can see a snapshot of the simulated structure of a protein that is interacting, not a protein, of an mRNA, where the triplet codon is forming interactions or forming bonds with the tRNA anticodon loop. And here you can see the hydrogen bonding that is taking place between the two. And as you can see, it is written here as well, this match has to be perfect so that only a proper tRNA is capable of the recognition and only the proper amino acid is getting incorporated into the chain. And one of the major players that makes sure that this incorporation is accurate or it is correct is the ribosome. So while I don't really want to get into much detail about it, you can read up on it because it's very interesting. Every time that the ribosome incorporates, so like I said, the ribosome shifts 3 bases at a time, right? So after it has shifted 3 bases and it has seen what is the mRNA sequence, and it has allowed incorporation of a correct tRNA in its like complementary to that sequence, the ribosome is said to nod. Basically, while it does not physically nod, there is a movement in the mRNA, movement in the ribosome large subunit, which appears to be like a nod, which basically means that the ribosome is now allowing translocation or allowing movement to proceed forward. So these are all very small details that are still being very extensively studied. So maybe we'll find out more about this in the future. But till now, this is known that the process of translocation only proceeds after the ribosome has approved that, yes, the incorporation was correct, and now we can move further. So that is the idea. Now, just now a student was mentioning, you can see that there is hydrogen bonding between the first and the second bases, which is um, mostly, more often than not, unless there is a mutation, it follows the Watson-Crick base pairing that we have discussed, and some amount of flexibility or some amount of uh, I wouldn't say error, but some amount of leeway is given to the third position, where you can see that it is said to be the wobble sequence because it is less constrained and some other bases might be allowed to come in, and therefore it is also considered to be a consequence of degeneracy as well. So the reason is that it is evolutionarily a conserved mechanism because it is allowing you to reduce the number of tRNA genes which the organism would need, and also for it is a sort of a mechanism that would protect against mutations as well. And it is allowed because of some stabilization by the tRNA and mRNA binding by the ribosomes. So that was the process. Now we have discussed in detail about the tRNA. This is just a snapshot of the process of protein synthesis. Let me just see if the other lectures have better detail. I don't think so. I think it's the same copy. So, okay, we'll look at this image only. So this is a snapshot of the process of protein synthesis, where you can see that represented in brown is the ribosome. You have the small subunit here, you have the larger subunit here. Within the ribosome, you will have the A, P, and E site. While the E site is not really clearly distinguished here, it is present. So what basically happens is that the ribosome is moving here towards the right and your mRNA sequence is being read. And as it is being read, first the correct tRNA containing the correct amino acid is uh, allowed to bind, and it is allowed to form these codon anticodon um, interactions or recognition. And once that recognition has been performed, it is allowing the incorporation of the correct amino acid, and a phosphodiester bond, not phosphodiester, sorry, peptide bond is created between these two proteins. And after this peptide bond has been formed, it is shifted onto the adjacent tRNA, and then that is allowed to be exited, or that is allowed to be released through the peptide exit tunnel, which is at the top of the ribosome. So these are the basic processes. So first recognition is between tRNA, anticodon and codon from the mRNA, which you can see here. After that recognition has, just give me a second. Yeah. So after the recognition between codon and anticodon has been carried out, the next step is that a peptide bond has to be formed between the now incorporated amino acid, and therefore the synthesized polypeptide chain is allowed to extend, and then it would, after it has been completely synthesized, it would be released from the ribosome, and then the ribosome subunits would disassemble, or they would detach from each other. There would be assembly for a new gene or a new mRNA that has to be translated at some other point of time. So that is kind of the idea that goes on here. I think this is the same lecture. Yeah. So here we see, till now we have discussed transcription, the genetic code, and the basic idea behind translation as well. Now comes the idea of Now comes the idea where proteins are then initially synthesized as a polypeptide chain, which is like a string of nucleotides. And later it folds into different shapes. So there are several stages in which a protein can fold. The first stage, or the least complicated stage, is known to be the primary protein structure, where you just have a continuous stretch or a chain of amino acids. Next is the secondary protein structure, which is the where some regions of the protein, they fold into these two distinct structure. One is the alpha helix, the other is the beta sheet. So these are two types of secondary structural motifs that you can find. These are extremely detailed. They've been studied in a lot of detail. You can refer to any of the reference books, or if you want suggestions, I can specifically recommend Brandon and Too's Protein Structure by Brandon and Too's to be one of the best books if you are interested in studying these in more detail. So a lot of chemistry goes behind each of these structures. Like there are individual hydrogen bonds which are present between these bases, which is why it assumes this specific helical structure, or why it is able to form a sheet. So that is a lot of detail, which is certainly beyond the scope of this course, but you can look it up. And if you want some information, I can help you out with it. But yeah, that is the idea. Then individually, these secondary structural motifs, they are able to interact with each other, and they're able to form 3D structures known as the tertiary protein structure. And these tertiary protein structure would then fold among themselves, or they would associate with each other to form the quaternary protein structure, which has multiple amino acid individually or separately folded amino acid chains, which assemble with each other to form the final protein. Like for example, if you take helicase, for example, helicase has 6 individual protein structures that have all assembled together to perform its function. Or if you take hemoglobin as an example, hemoglobin has 4 separate subunits, each of which have assembled to form the quaternary protein structure. I think we could say, if we take an example from today's class only, RNA polymerase, we saw that it had multiple subunits. It had alpha, beta, sigma, etc., many different subunits. And these also have assembled to form the quaternary functional protein structure. So here you can see that this is the protein data bank. The idea about PDB, or protein data bank, has been explored here. It is a repository for all the different types of protein structure that are being studied and are still being studied. Whenever you discover a protein structure, its function, localization, anything about it, any information about it is always stored within the PDB, within this protein data bank. And there are multiple such repositories available on the internet as well. And you can go and explore it to your heart's content as much as you want. Here are some examples of different ways in which proteins can fold, like here you see GFP or green fluorescent protein, which forms the beta sheet or beta barrel structure that we discussed. APF1, you can see some histones which are present. If we discussed histones in the last class, where we talked about how there is an octamer, which means 8 proteins at the center, and DNA is wrapped around it. If you remember, we had an analogy of a computer mouse and the wire wrapped around it as an example. So equivalent to that, computer mouse is basically, if it was made up of 8 proteins, that is the analogy you would be able to see here. And this is another specific type of structure where you can see that there are one example of this. This is commonly known as the horseshoe motif because it looks like a horseshoe, and it has alpha helices on the outside, and it has beta strands on the inside, and it forms this very distinct structure, which is necessary for it to perform its function. So that is what you see here. Um, Next, coming to the amino acids that we have discussed previously, you can see that they can be subcategorized into multiple different classes like non-polar, polar, their charge, their acidic or basic nature, etc. And some of them have some special properties, like you can see here, for example, the disulfide linkages, which can be formed between amino acids that contain sulfur, and mostly these are formed between cysteines, two different cysteines represented by C. Like here you can see C, which has CH2SH as the side chain. These two sulfur groups, they are able to form a bond between each other, and they are one of the major contributors towards protein structure, as they help in, like you can see, they can help a straight chain fold into sort of a loop-like structure, and therefore they are one of the most stable and one of the most common, not very common, but one of the most important contributors to 3D protein structure. And you can also see proline, glycine here has been indicated. So we come to the, since we are talking about structure here, the peptide backbone, the polypeptide backbone has a certain arrangement, and there is a concept of dihedral angles also which comes in. So um, in case of DNA, we had discussed its backbone as well. Its backbone is completely different. Its backbone is a phosphodiester backbone where you have phosphate moieties which are being linked to each other, and the sugar and base are towards the inside. So in case of proteins, however, the backbone is made up of peptide bonds that we have just discussed, right, during the process of translation, where individual amino acids are forming peptide bonds with each other. So you can see here that they form CO and C double bond O and NH. This CONH moiety is known as the peptide bond. Other than that, the carbon molecules that you see, those are alpha carbons, which are at the center of each of the amino acids. And what you see projected towards the outside are the side chains. R1, R2, R3, etc. are side chains, which can vary from one amino acid to the next. Now, coming to the concept of the dihedral angles, there's a better representation later, we'll come to it. But dihedral angles are basically um, the angles that arise between two different, they are torsion angles or dihedral angles, which arise when you consider the three-dimensional structure of a protein. So when a protein is rotating, or when the amino acids within a protein, they are forming certain angles with each other because of rotation. You can see that here you can see some arrows which are indicating rotation. So when they orient themselves in a certain manner, only certain angles are allowed, certain angles are not allowed. What do we mean by this allowance or not allowance? So this comes because each amino acid has a distinct side chain, and these side chains are also individually made up of certain atoms. These atoms have electrons, right, in their outermost shell. So when two atoms from two different side chains are very close to each other, there is repulsion between the two, and that structure is never stable. Therefore, such type of hindrance or such type of obstacles are known as steric hindrance. So only certain angles or certain orientations between different amino acids are allowed, and these allowed and disallowed regions are understood via a Ramachandran plot, which we'll come to next. So if it is, I hope it's not very confusing, but yeah, these were all part of the lecture, so I am going through it a little bit fast. So here you see the alpha helix and beta sheet or beta turn, which I just mentioned, and you can see how it is forming interactions with, like in this case, it is a DNA binding protein, so how it is able to interact with the DNA is an example you can see here. And you can see that tertiary structure usually has mixtures of these proteins. So there are alpha helices here, and there is a beta sheet in between as well, which forms some distinct folds, like Rossmann fold, some sandwiches, some barrels, etc. All of these different types of motifs or different types of tertiary structures can be studied. Again, these are very individual topics, like these can be entire courses by themselves because there is so much information on it. For your curiosity, if you want, please go ahead and read more. But for now, this information is good enough. So here you can see some, like it's mentioned here, some spectacular protein structures. You can see how it has how a beta sheet is formed with respect to each other. You can see alpha helices. You can see that hydrogen bonds are one of the major contributors that help in stabilization of these structures. And here you see that there are different representation of protein structures as well. So most commonly what is used is the ribbon diagram, so that like this is called the ribbon diagram because if you look at the alpha helices, they are as if you three-dimensionally try to position a ribbon in a certain way, you will get this orientation. So it is one of the most preferred representations because it allows you to look at the possible interactions between amino acid side chains. If you remember the structure of the ribosome that also we had initially discussed, then that also was a ribbon diagram which was used for representation. Other than that, we have space filling and surface diagrams as well, which can be used. So yeah, these are the torsion angles that I was just talking about. That I was talking about. So the two major torsion angles or dihedral angles are phi and psi, and the third is omega. So here you can see the 3D representation for that. So the phi angle is said to be between N and C alpha. C alpha is of course contributed by the amino acid, and N is part of the peptide bond. So here you can see C alpha, and this is N or nitrogen. The angle between these two is known as phi, whereas the angle between C alpha and C are psi. So the angle that they form upon rotation with respect to each other, that is denoted by these dihedral angles. And omega, on the other hand, is between C prime and N. So C prime is part of the side chain and not side chain, the part of the peptide bond, and the angle that it forms with the adjacent nitrogen, that is known as C omega. So each of these angles are studied in great detail. And like I said, this again was another extremely relevant discovery to the field of structural biology, which was the Ramachandran map. Again, a discovery by or a contribution by an Indian scientist, where he, in the form of a sort of a plot, he represented what are the allowed and disallowed regions of proteins, where he studied a very large number of proteins and he saw what are the allowed angles. So he represented phi and psi from -180 to +180, which is the range that is used for these angles. Again, this is a very common question for competitive exams. Please go through that. So he plotted these two against each other, and he saw what are the allowed regions, and among them, some common regions were studied in more detail, like you can see here, for example, right-handed alpha helix, left-handed alpha helix, polyproline 2, beta sheets, beta turns, 310 helix, pi helix, etc. So they allowed a very clear understanding of how exactly or what exactly are the possible conformations that a protein can ultimately take in 3 dimensions, but he represented that in a 2-dimensional chart. So again, a landmark discovery, like I said. So here you see this is Dr. G.N. Ramachandran, who is credited with this incredible discovery. And you can read these papers. These might be slightly beyond the scope of this course, might be a little difficult, but you can certainly go ahead and explore it. And yeah, like you can see, these were incredible publications that were made in Nature and as early as 1954, 1955. So excellent discovery by C. Ramakrishnan and Sai Sekharan. All of them made very large contributions to this whole discovery. So following that, let me see. This is so of course. This is Yeah, so here is a representation of the amount of information that has come in the last few years, especially after bioinformatics and tools for visualization of protein structure are as technology has advanced, the understanding of protein structure has increased exponentially. So you can see here that these are the different methods that are used for understanding protein structure. There's X-ray diffraction, NMR, electron microscopy, and hybrid methods, which use all of them. And their utility has been very, very extensively explored to understand protein structure, and they have been shown in multiple publications as well. So that is certainly a major contributor to what we know about proteins till now. So guys, give it some. Yeah. So again, these are the amino acid structures that you can see, the dihedral angles that we have discussed. The structure of the peptide bond has also been explored here, where there are multiple features of the peptide bond. It's not just this much. You can again go into it. Like the individual angles are defined, but for now, what you can know is that the peptide bond can have cis and trans orientation based on how the individual amino acids are present. And like you can see the cis orientation, like I said, because of the presence of electrons in the outer valence shell, there can be a lot of repulsion when subunits are arranged in a cis orientation versus oops, versus when they are in a trans orientation, they are more relaxed, there is less repulsion, and therefore, under most circumstances, it has a lot less steric hindrance. So these are the individual bonds, bond angles, and bond lengths in the polypeptide chain for the peptide bond. So you can go through those as well. You don't need to remember all of it. You can just go for its understanding. You can remember some, like for example, the CC bond or the individual CONH bonds, but certainly go through it as much as you can. Uh, then the different types of hydrogen bonds that are found in all these different types of molecules, they have also been explored here. So the different types of donors and acceptors, the kind of bonds they can form, then the polar nature of covalent bond, the hydrogen bond comparisons can be made. So I think somebody had asked this the last day as well, that why is hydrogen bond that relevant? So you can see that majority of life's biological macromolecules are very, very heavily dependent upon the formation and the stability of hydrogen bonding in their case. So these are just some examples for it, and you can see, you can read up on it even further if you want more detail. Um, like here you can see other representations of the Ramachandran plot. You can read up on its significance. You can read up how it was discovered, how long it took. You'll find all of these details. But the basic purpose of it was to get an idea about the allowed and disallowed dihedral angles pi and psi within a 3D protein structure. So yeah, now there are two major exceptions to the Ramachandran plot as well. For example, glycine, which has extensive conformational flexibility, which means that it can be present anywhere within the Ramachandran plot. And if you just try to understand it very logically, then you would see that because of its structure, um, because of the fact that its side chain is just a hydrogen moiety, it does not have much really occluding it. Therefore, it has such incredible conformational flexibility. And similarly, we can see another exception, which is for proline, but it has a completely separate set of rules. That is because it has a very, very constrained structure. It does not have a main chain. It does not have the same number of atoms available as that of others. And because it forms a ring upon itself, therefore it has very, very few structures or very, very few orientations that are allowed, and therefore it is the most restricted amino acid that is present among the 20 that we have studied. So yeah, that kind of gives the idea for the same. Now coming to the last part that we'll be taking up today, which is protein folding, and we'll try to wrap this up quickly, and then we can move on to the questions so that we can end the class on time. So till now, if we just go over what has been discussed, we talked about the process of we talked about the central dogma replication previously. After DNA is replicated, it is getting converted or getting transcribed into RNA, particularly mRNA for protein coding genes. Then that mRNA is undergoing the process of translation by ribosomes, and then that ribosome is releasing the synthesized polypeptide. That polypeptide can then assume a structure which will allow it to perform its function. The question is, how is it attaining that structure? So it does so by a process known as protein folding. And what needs to be one of the major things that need to be ensured is that the protein must fold in, or the structure must be formed in such a way that it is stable. So that is what we look at here basically. So you see here, there is a U state, which is the unstructured state, or the normal state of, or unfolded state of the protein, followed by an intermediate state where we see that there are some regions within the protein which start to form a sort of start to form a 3D structure. And majorly we can see that in the form of some alpha helices, because those are usually the first regions of a protein that take up their 3D structure. So that is the intermediate state. And then finally, in the third state, we can see that they form the native or the functionally relevant form of that same protein. So this is the problem with, not the problem, the current status of protein folding is such that um, there are multiple theories, there are multiple concepts which are being explored by scientists, but there is no defined or unified opinion about the process of protein folding. There are many ways in which it might happen. So we will not get into the nitty-gritty of it. We will just try to understand it from a bit of a distance. So basically, the idea behind it is that you want to get a structure which is the most, how do I put it, the most stable structure. When we mean stable, it is with respect to thermodynamic stability, which means it should have the least amount of entropy possible, but it should also be capable of performing its function. So therefore, this concept of stability is brought about by a number of features, which includes the fact that the because the environment around a protein within a cell is the cytoplasm. It is basically the cytoplasm, which is an aqueous environment. Therefore, what the protein tries to do is that it tries to minimize the number of amino acids which are sort of hydrophobic in nature. It tries to minimize their exposure to water or their exposure to the aqueous environment by bringing them to the center of the protein. So this concept is basically building of a hydrophobicity core. So water, or the presence of water around the protein, is the major contributor to this. Other than that, you have a number of interactions like formation of salt bridges, which are ionic interactions. You have hydrogen bonds, you have dipole-dipole interactions, you have disulfide linkage formation between cysteine moieties that I talked about previously. You have some metals which can bind in certain proteins. For example, polymerases, they usually involve the function of a divalent cation. Most polymerases, they need such divalent cations such as magnesium, calcium, etc., which serve as cofactors for whatever process they are trying to carry out. So those metal binding sites are also important for contributing towards protein stability. And finally, the objective is to reduce the unfolded state entropy to bring the final protein structure. So here you have, so I'll not get into this. I think it was explained in the lecture as well, the thermodynamic factors that contribute towards the final stability of the protein. Then came the protein folding problem, whereby Leventhal had initially given an idea that if really protein folding has to occur one amino acid or one region at a time, then he gave an example of a polypeptide where there are 100 residues needing 99 peptide bonds and all of the different possible phi-psi bond angles. So if it had to sample every possible conformation, then one protein folding event would take exceptionally long, and therefore attaining that structure would be a very, very long process. And that is certainly not something that happens inside the cell. Uh, it is a matter of some few seconds, which ultimately gives you these ultimately functional proteins. So that was a problem that he talked about. Then what was talked about was that the relationship between the sequence that you have. So if you are just given one sequence, and because all of the different possible combinations or orientations or foci angles which can be allowed are true for every sequence, we have to make sure that every time that protein is synthesized from that particular mRNA sequence, every time the protein remains the same. So it must attain that unique fold, and it must be capable of performing that function, and that is ensured by the thermodynamic stability. So the reason it is called the protein folding problem is because at that point of time, even to a large extent today, people did not completely understand the protein folding problem, that how is one sequence giving us one particular structure, and how is it doing it so fast under the time scale that we see inside the cell. So these are very, very crucial topics of research which are being taken up even today. Then you see the contributions of Anfinsen, Moore, and Steen, where they understood the structure of ribonuclease A, and they saw that when they initially they took ribonuclease, they added some reducing agent, which was getting rid of the disulfide linkages within that protein, RNase A, which has four separate disulfide linkages, some of the largest which are known to mankind till date. And then they added urea to it, which is a denaturing agent. They obtained unfolded ribonuclease, but when they removed that urea, when they removed the heat aspect, it was able to get back its initially functional structure. So they kind of understood that the protein folding problem proceeds via some sort of intermediate stages, it where it narrows down the number of possible conformations, the number of possible 3D structures that it can go through to ultimately arrive at its unique or its functional state. So that is what was explored by Anfinsen, and not just by Anfinsen, but several other scientists as well. And like we said, hydrophobicity is one of the major contributors to this process. And apart from that, the thermodynamic stability for the same is also extensively explored. So if we just look at the thermodynamics of protein folding, you can see that it is represented by the equation del G, which is the Gibbs free energy, which is also giving you an idea of how stable or how much movement or how stable a particular entity is inside the cell, how thermodynamically stable it is. And you have enthalpy and entropy as the two major contributors for it. So you can see that the during protein folding, the contributions to delta G or Gibbs free energy are made by hydrophobic effects, hydrogen bonds, and van der Waals interaction, which try to reduce it. On the other hand, you can see that the chain conformation entropy, it is increasing it. So net effectively, the delta G becomes less than 0, which makes the final protein structure quite stable. So this you can look at the individual contributing factors in extensive detail if you're interested in the same. And here you can see that these are the possible hypotheses which are being looked at, that there is a protein folding funnel. You can see that it goes through, I don't know if it is detailed here, but basically what it does is what is speculated is that every protein during the process of folding goes through multiple energy minimas. It tries to fold in multiple conformations, but any structure that is not thermodynamically very stable, it will immediately try to get out of that protein folding local minima, and will try to refold again before it arrives at the global minima, and then that structure becomes the most feasible or the most acceptable to the cell, and therefore that is allowed to continue and perform its function. So yeah, basically that is the hypothesis which is being explored till now, till date. So I think these also have the same slides. Mostly, yeah. So we can go towards the questions. So these were majorly the topics that were looked at in week 2. And what we will do is we'll look at some sample questions. So that would probably help you with your assignment, and then we can get into details further in the next week. So here we were asked which of the following codons initiates the translation of mRNA. So like you already have studied that methionine, or in further detail, formyl methionine is one of the major reasons that at all ribosome is able to initiate translation, and methionine is encoded for by AUG, which is the triplet code for it. So that would be the correct answer in this case. The other UUU is for phenylalanine. UAG is for a stop codon. So these are not correct answers for the same. Then in question 2, we were asked that which of the following is a property of introns in eukaryotic pre-mRNA? So make sure you look at the terminologies carefully. You have eukaryotic pre-mRNA, and you have the presence, obviously you have the presence of intron because it is eukaryotic. So you're being asked about introns in general. So they are, which of the following is true? They are non-coding sequences that separate exons in monocistronic pre-mRNAs. The answer is true. They are palindromic sequences that self-splice to form lariat structures. No, that is false. That is something that this lariat structure formation is something that occurs in case of splicing. Then there are extra coding sequences which can be removed by an RNA protein complex. They are extra sequences, but they're not coding in nature, introns. So this is also not true. They can remain in mature mRNA due to alternate splicing pathways. Instead, it is completely the opposite, because no, even in alternate splicing, what happens is that different combinations of exons are allowed to attach to each other. But in case of any sort of splicing, introns are not retained, or they should not be retained. So the correct answer for this would be the first one. Then the next question was about remdesivir, which most of you might have heard of, especially if you were familiar with the situation during the pandemic. So remdesivir was an antiviral medicine that was approved for use in many cases of emergency, particularly for COVID-19. The question is that which step related to viral replication represented in the figure below does remdesivir target? So basically what the objective of it was to stop the propagation of COVID-19. And if you remember, coronavirus had an RNA genome, and therefore what it was trying to do was that it was to stop that replication process. So if you look at the central dogma that was indicated there, the correct option for it would correspond to the step that shows duplication of RNA or replication of RNA. So that is being indicated by step number 2. So that would be the correct option for this. Then we're being asked about transcription. So during transcription, which strands of dash are used as template? So quite an easy question. So if you talk about the molecule which is being used, then that is certainly DNA, because DNA is used as a template for synthesis of mRNA. And if we talk about the strand or strands which are being used, the number of them, then we know that certainly one strand only is used as a template, and that only undergoes the process of transcription. So that is the idea behind that, is that you are effectively getting the same sequence. So there is the coding strand and the non-coding strand, and the non-coding strand is being used as the template, while the other strand is basically the code that the synthesized mRNA will carry forward. Here we are being asked that the formation of the lariat-like structure, this is something I just discussed as well. The lariat-like structure in the electron microscopy image below is characteristic of the process of A, replication, B, splicing, C, transcription, or D, protein synthesis. So if you look carefully at the structure, you'll be able to see that there is some sort of a loop which is present here in the form of a lariat, and that is indicative of the process of splicing. Replication can also be seen in electron microscopy images, but that's a different way. You can look it up. The theta mode of replication can often be seen, where you can see the replication bubble that is present within the within the image. And transcription or protein synthesis are extremely difficult to visualize. You can see some extent of it, but they're quite difficult to actually visualize by electron microscopy. You can, but yeah, in those days it was not feasible, and this is representative of splicing only. Um, then in question 6, we are asked that because there are 4 is to the power 3, you are getting 64 possible combinations of triplet codons. Most amino acids are specified for by more than one codon. So this is the concept of um, yeah. So this is the concept of degeneracy, like we have discussed, that more than the number of amino acids versus the number of possible codons that are present, they do not tally with each other. And that is the reason why multiple codons are encoding for the same amino acid, uh, giving about the degeneracy of the genetic code. Then in question 7, we are asked that which of the following amino acids are an exception to the Ramachandran plot? Um, we also saw this, it was there directly in the lectures. So there is proline, alanine, or glycine or lysine majorly as the options. And the answer to that, since we already know, I'm getting directly to it. It is glycine and proline, which are exceptions to the same. Glycine because of its extreme conformational flexibility, and proline because of its very, very restricted structure. So that is what you can see as the correct option here as well. In question 8, we are asked which of the following protein structures can be predicted with the help of the Ramachandran plot. So if you remember the plot images that were presented before, you can see that there are some specific regions which are attributed to some specific secondary structures. So in that case, only some regions can be attributed to have alpha helices, some have left-handed, some have right-handed alpha helices, some have beta sheets, beta turns, etc. So the secondary structures of proteins can be quite easily predicted with the help of the Ramachandran plot. And the last 2 questions are about the omega angle of the peptide bond, which is like if you remember, the phi-psi angles are the dihedral angles, where the omega angle was between the uh, N and the C prime, which is basically part of the peptide bond only. So the omega angle is mostly planar. So it's an indirect way of asking you the exact question that what is the nature of the planar bond? So of the peptide bond. So the peptide bond is planar, and because the omega angle is equivalent to the angle between the peptide bond contributing moieties, therefore the answer will be planar. And in the last question, this is again what we discussed during the Ramachandran plot as well. The rotational span, or the range in which phi and psi angles can vary, that is from which of the following? So the answer to that is from -180 to +180. Basically, it is covering all possible orientations, 360 degrees, but its representation is always done in this manner, that it is from -180 to +180. And based on that, the entire plot has been made. So these were the sample questions for this week. If you have any questions, please do let me know. And apart from that, in this week's slides, I have also attached a link for some information on prion diseases. I think that was brought about in the last week regarding their transmission. So I read up a little bit on it. You can go to the same link and check it out for your own understanding as well. So basically what has till now really been understood about prion diseases is that while they are considered to be transmissible, the transmission is not like that of other pathogens. Like if somebody sneezes, you are not going to get that same viral infection, like in case of COVID, or somebody's coughing or sneezing. It's not um, it's transmissible, but it's not contagious. They are different. Contagious is what you get from touching or coming in contact with other people. Uh, the most common mode of transmission for such prion diseases was usually by consumption of meat. So it was particularly very evident during in certain cultures in very early, like I'm talking about many thousands of years ago, when cannibalism was a culture that was often seen in some certain populations. So an infected person was capable of transmitting those proteins to other people, and then they would suffer from the same disease. Or if somebody ends up consuming some sort of meat, like in a lot of cultures you can see that people they consume, not just cultures, in a lot of populations you can see people consuming animals which are often infected with such diseases. For example, scrapie that we were discussing in the last class, it infects cows. So people who consume cow meat as part of their diet can also often end up contracting such diseases, and that is considered to be the major mode of transmission. However, I could not find any reports about any nucleic acid being a contributor to it. In fact, a lot of studies were done on it as well. So mostly when there is a nucleic acid which is a contributor, you can usually inactivate that particular disease causing agent using UV light. Basically, UV light is damaging the DNA, damaging the DNA or RNA, and therefore that organism is no longer capable of surviving. However, they were not able to do this with these group of disease causing agents, and therefore it was concluded that they do not have a nucleic acid intermediate, and they are just proteins which are able to carry out this function. So while there are still, this is only leading to more questions than answers, this is still a field which I believe is being studied, and hopefully we'll have some answers sometime in the future. So that was all I had to present for this class. If you have any questions, please go ahead, and then we can wrap up for today. Okay, if there are no more questions, you can certainly drop more questions if you have any in the portal that you get on the NPTEL website, and somebody will get back to you. But if not anymore, then thank you for joining today. I hope your doubts were clarified. We will again meet for another session in the next week. So thank you and good night.
Ma'am, class over.
Yeah.